you for coming. Thank you for having me. When Dr. Liu asked me to talk about diagnostics today and putting it in the context of micro and nanoscale technologies, I decided to choose one specific technology we've been developing that isn't quite yet at the micro scale, but I wanted to talk to you about it anyway because we're pretty excited about it. I just want to give you a quick overview of what our lab does. So in addition to being a bioengineer, I'm a pediatric hematologist oncologist as well, based over at Emory, and really our lab focuses on cell mechanics research, specifically in blood and cancer. So we do quite a bit of collaborations with the other speakers that we're actually sharing the stage here with Dr. SelfCheck and with TJ and Dr. Val as well. And we use similar systems that Dr. Liu, Dr. SelfCheck talked about to really allow us to answer questions that were technologically infeasible before. But we're also interested in turning these devices and systems we develop into a diagnostic and bringing it back to my patients that I see, and then really fulfilling this circle. So the one thing I'm going to talk to you about today is this new diagnostic that we're developing in the lab that we call the AnimoCheck. And I think this project really highlights the quality and opportunities we have over here at Georgia Tech, because this device really started as an undergrad project that I helped mentor, and it's just grown and accelerated, and it's gotten to the point where it's become a major part of our laboratory. And the fact that we have these capabilities here to train such great undergrads is something I've been very amazed and very pleased by. So a little background about what this is. So this project, as you might gather from the name, has to do with anemia. And anemia, by definition, means low hemoglobin levels, and it affects a lot of people. Worldwide, 1.6 billion people are affected. Major consequences for not only health, because so many people have this disease, it affects social and economic development as well. Occurs in all stages of life. Women, especially those of pregnancy age, and young children are most likely to be affected. The nutritional deficiencies, such as iron, you guys have all heard of that. Iron deficiency anemia is actually the most common cause. Zooming in a little bit about the demographics that are involved. So millions of people are at risk, even in the US here, right? The elderly, as I mentioned, pregnant females, patients who are on dialysis, kidney failure patients are anemic, patients who have cancer on chemotherapy, radiation therapy, as a side effect of their medications are anemic. And then there are patients with chronic anemias, and even diseases that affect the immune system can cause anemia as well. And I'm going to talk a little bit about some of these demographics. So children, very young children are always at risk for anemia because of their development. Their iron needs are greater than in other times of life. So right now, the American Academy of Pediatrics currently advises that every child at around 1 year of age actually gets a blood count to screen for anemia. Then we have these patients who have chronic anemia. There are diseases in which the bone marrow is not making red blood cells, or there are diseases in which the red blood cells are being actively destroyed. Immune system diseases can cause anemia as well. So lupus, HIV, rheumatoid arthritis, all fairly large demographics here, even in this country. These patients all have chronic anemia. And I mentioned to you before the kidney disease patients and the cancer patients. So right now, these patients all require frequent monitoring of their blood counts to assess their level of hemoglobin. And often patients can develop these acute, severe bouts of their anemia that could be life-threatening. So if you look at the rest of the world, this is an even bigger problem. In fact, in developing countries, anemia affects over 40% of young children and women. The big three global threats that the WHO defined, HIV, tuberculosis, malaria, all cause anemia. And specifically in malaria, it's these acute episodes of anemia that are a major cause of mortality. And because of the lack of medical resources, diagnosing and screening for anemia is actually cost-prohibitive. So it's somewhat ironic that where you need it the most, you actually can't have the diagnostic. And what is the diagnostic gold standard? Well, it's something called a blood count, CBC, the most commonly used medical test, period. So all of you have likely had a CBC. in the past. The current CBC systems are hospital-based, and they're fairly expensive, as you can see here. They require a technician, a lot of moving parts, electronic parts, currently based in clinics and hospitals, not for home use, and is fairly complex in terms of its use. There are now newer technologies that are point of care anemia diagnostics, but they have some limitations as well. So there are some portable ones that exist, and we currently see them in clinical settings such as ERs, clinics, operating rooms, where a device that looks like this little handheld thing can, if you insert a cuvette with a blood sample, read the hemoglobin level, i.e. how anemic you are. There are newer devices that are non-invasive. This company has just come out in the last couple of years with a portable device that works similar to a pulse oximeter, if you guys have heard that, where it's a cutaneous read and one can obtain a hemoglobin value. Expensive, and newer studies have only shown that that's reliable only in certain clinical contexts, and it is used in certain settings, again, most likely in the emergency room. Anesthesiologists use it in the operating room, and the key, though, is that they are all fairly expensive and require additional components, not suited as a home-based point-of-care self-test, which is really the goal of our project. So what we have developed is this simple color-based test for hemoglobin levels in which a single drop of whole blood leads to a very easy visual readout, right? And we are targeting it to be used for all ages and subgroups, and really we're trying to make it as safe to use as possible, simple. Currently, this assay involves a redox reaction with the hemoglobin in the blood. It takes less than a minute, and we think it works basically the same type of use model as a diabetic would with a diabetic glucometer. In addition, we're excited about it because you don't even need that additional piece of equipment. You don't need the glucometer. There's just a color scale that you can read. So how does it work? It's fairly simple right now. So really it takes two components. One is a capillary blood collection tube, and then another larger tube that's pre-filled with our reagents. So the way the patient would use it would be a small finger stick with a pain-free needle that are available commercially now. 5 microliters of blood is all you need. Insert it into the tube, wait, shake, and then you interpret color. So here is a demo. Drop of blood, wick it in, and you fill up the tube. Put it in the reagent tube, and it looks like Erica painted her nails to match her gloves that day. Shake it up. Make a sound. And you get this color. So our results are basically as follows. If you're in the healthy range, you get this reddish to orange, and as you get more and more anemic, it becomes more so you have this range of about 5 colors, and currently we're trying to determine what the resolution is. But certainly we can at least, with the current assay, say you're healthy, you're mildly anemic, you're moderately anemic, you're severely anemic. So how do you quantify it? Well, using a simple spectrophotometry, you can actually get quantifiable information. So what you see here is inserting our assay with results of typical hemoglobin levels. So normal is about 16, and as you get lower and lower, fairly anemic is around 8. You see these different curves, and at different wavelengths you get these different peaks. What is interesting here is that there are 2 select wavelengths where the different levels seem to have different peaks and dips. So what we do is we take the intensity of those 2 wavelengths and we come up with a ratio. And then if you look at the ratio of these different samples of anemia, you get this nice correlation. So therefore, we can correlate specific color with a level of hemoglobin. Now we're trying to characterize it. For example, one question we have is, well, do substances in the blood interfere, right? If this is a color-based assay, sometimes there are things that may occur in the blood that give off a certain color. How does that interfere? So one common thing would be fat, right? a lot of people with high cholesterol, I've actually seen these blood samples actually have a grayish hue to their blood. Does this interfere? Well, we're doing some studies right now, and here's a study in which we infuse something called intralipid, which is kind of an artificial lipid that patients can use right now if they can't eat. This could be infused intravenously, just to see if this lipid at least interfered with our assay, and it certainly doesn't seem like it. We're currently assessing other substances as well, bilirubin, other products of hemolysis. Then we actually went straight ahead and did a preclinical assessment. We went to our hematopathology bank lab and just took samples of known blood counts. The one problem is that these samples were obtained in tubes that have an anticoagulant that prevents the blood from clotting in the tube, called EDTA, and that is known to interfere with our reaction. So you do get this variance that we would like to see go away, but you do see this general trend with this large number of patient samples that in general, the more anemic you are, the more blue you are, the more healthy you are, the closer you are to red. So certainly there's these limitations. So really what that posed to us is we need to do a more formal controlled clinical assessment study. So ongoing efforts right now are we're really characterizing our assay from a biochemical standpoint. And then, and here's the micro part, prototyping optimization. A big chunk of our lab does a lot of microfluidics, as Dr. Liu had said. And certainly we think one way to make things even simpler is to come up with a microfluidic interface that can really optimize the usability on the patient side. So we're currently starting a clinical assessment of this system, using this system compared to a standard blood count, and we're leveraging all our resources both over at Children's Healthcare Atlanta, as well as on the adult side over at Emory's Winship Cancer Center. And we expect to be done in about 12 months, and we're pretty excited about this test, and we will certainly have hopefully a publication within a year or so. So how would we actually envision this being used? I kind of alluded to this in the very beginning, but with the system, in theory, anyone could screen themselves for anemia, children or adults, and for the subset of people who are chronically anemic, they could actually self-monitor their own anemia and hopefully be able to catch the severe episodes that they can have, their acute episodes of severe anemia, before things actually take a turn for the worse. And then what I'm most excited about is the fact that theoretically this could be an inexpensive alternative to blood counts in resource poor or global health settings. Here's our funding. We couldn't have done this without funding from a lot of local resources. The Georgia Research Alliance liked our project enough for them to actually fund us for the last few months, and the FDA grant that we have here that is used to really facilitate the commercialization of pediatric medical devices here at the Atlanta Pediatric Device Consortium. Andrew Lyon over at Georgia Tech here, who's been our resident chemist, and our colleagues over at Emory show up. So with that, I'll take any questions. Thank you.
Do you have access to all the data on those patients so you can see if there's other correlating factors? Or?
Not with the existing patients, but certainly for our forward clinical assessment, we'll be looking at that.
What's the impact of time and temperature on the assay?
Yeah, so it's a great question. And so what we're going to be doing in terms of assay characterization. So right now, time we think is pretty good. We've had samples of our assay for at least just stored for a couple months, and they still seem to work. Light they're sensitive to light, so we do have to shield it in simple foil. Seems to be okay. The temperature stuff is currently what we're working on right now. It does seem that if you get above 40 degrees, it does interfere a bit. So we're trying to A, get a better sense of just what the parameter space is, and then B, see if we can optimize that.
What's the mechanism for the color change, and can it be applied to other things in the CBC?
Yeah, so it's a redox reaction that is that involves hemoglobin as a catalyst for a peroxidase reaction, essentially. And can it be used for other things? Yeah, theoretically, I mean, one could use it to look at, for example, the different types of hemoglobin diseases and so forth. Having said that, there's other assays that can do that as well. Yes, ma'am.
So I thought when I went to give blood, they took a prick from my finger and put a droplet in something of appropriate density, and that told me if I'm anemic or not.
Yep.
So what's wrong with that very simple test?
That very simple test, requires another piece of equipment, essentially. What they most likely did is spun it down in a very small centrifuge, and it still takes a few minutes. So the goal here is to have it be completely self-contained, no equipment at all. All right, thank you.